Okay, so I think I'm going to make a start, and if anyone else wants to join, they can. So this is the session on neurodevelopment and neuroembryology. So I'm going to be giving this session. So this is a massive topic, and it's quite difficult to cover all of it. So what I'm going to do is I'm going to try and to kind of give you basic principles to approach both neuroanatomy and neuroembryology. And based off those principles, you guys can do further reading. What I'm also happy to do is kind of take questions as we go along, especially for the neuroanatomy section, because there's a lot of it. And I know it can get very confusing very quickly. So if you guys just kind of like write stuff in the chat, I can try my best to like keep an eye on it. It might be easier if you unmute just because it's hard to see the chat when I'm on, like sharing my screen on Zoom. But yeah, if not, you can ask at the end as well. So the more like you engage, the easier it will be as we go through this. So if we start, so I'm just going to begin by explaining what we're going to cover today. So like I said, it's neuroanatomy in particular is just so vast, there's so much of it to cover. So I just wanted to kind of give you basic principles that you can use to build on further knowledge. I'm going to try to cater as much as possible for the essays as well as the MCQs. So things that I think are high yield that are likely to come up. So we're going to start off by looking at just the anatomy of the skull, the cerebrum, and the brainstem. Then we're going to briefly focus on the ventricular system. I'm also going to try to bring in some clinical context as well. So that will make life easier, especially as you get into the clinical years, but also like gives you things to think about and maybe write about in your essays as well. So in terms of neurodevelopment, I've tried to break it down into four key principles. There's obviously lots of stuff about what structure becomes what. I think that's I think it's honestly easiest to just memorize that. So I've got like some tables and stuff from different people's notes and things that kind of make it clear what structure develops from what, which is going to be your MCQ questions. If you do the essay on neurodevelopment, I think if you break it down into key principles and think about it that way, you will tend to score highly. So I'm not going to really cover much of the spinal cord anatomy or development. A lot of the other lectures look at stuff like the sensory pathways and the motor pathways. So a lot of that will be covered in those lectures. So if no one has any questions, I think we can just kind of move on. Okay. Yeah, there's nothing in the chat. Okay. So I'm going to start off by thinking about the skull. So the skull is basically the bone around your brain. So the skull is comprised of 8 bones in relation to the actual cranial portion, and then you've got 14 bones around your face. So the image on the right shows you the different bones. There's the core 8 bones are the occipital bone, which is in the back, the temporal bones, which are in the side, the parietal bone, which is the sort of big one also on the side and extending into the back. And then you've got the sphenoid, which kind of pokes out there on the, sorry, can any of you see my mouse? I just want to understand if you can see that before I use it to point out things. If someone could maybe write that in the chat or something.
Yeah.
Oh, you can see my mouse. Okay, that's helpful. Thank you. So yeah, so the red portion has the sphenoid bone, which is basically which has 2 parts to it as well. And then there's the ethmoid, which is quite a small bone near the ear, just on the inside of the eye of the orbital groove. And then you've got the frontal bone, which is in the front. So the frontal, parietal, and the greater wing of the sphenoid, as well as the temporal bone, meet at the terion. So that's near your temple. So here you can see where the terion meets. So the terion is basically a weakness in the skull, and it's the weakest portion of the skull. So usually this area is located just around the temple, which is why if you have like a trauma or injury to the temple, you're much more likely to have issues and you're more likely to damage your brain. The real significance of the terion, though, also is that's where the middle meningeal artery passes. So this is a really common MCQ question you can get, which is which artery is going to be affected if you have damage to the terion. So this is definitely like something to write down and think about. So yeah, that's something to be aware of. So the next thing to think about is if you actually sort of open up the skull and look inside, you've got 3 main compartments to it. You've got the anterior cranial fossa, which is where sort of the frontal lobe of the brain sits. You've got the middle cranial fossa, which is where the temporal and parts of the parietal sit. And then you've got the posterior cranial fossa, which is where the cerebellum sits as well. So then the main hole that all of the structures that extend into the spinal cord pass through is called the foramen magnum. So yeah, you can look at all the images in your own time. I'm sorry, this is all going to be very, very fast-paced because I want to get it done in an hour. So then the next thing we're going to think about is the meninges, which are basically the layers around the brain. So the meninges has three layers. There's the dura, there's the arachnoid mater, and the pia. So the dura forms three sort of key structures around the skull that allows us to divide up different portions of the brain. There's the falx cerebri, which runs along the main portion in the middle of the brain, basically. There's the cerebelli, which is just divides the cerebrum from the cerebellum. And then there's the tentorum cerebelli as well, which does a similar sort of role. So that's just to kind of think about how you orientate yourself in the skull. So the next part is to think about the actual layers. So you have the outer, so the dura itself can be divided into two portions. So you've got the outer periosteal dura, which is continuous with the actual skull, the bone itself. Then on the inside, you've also got the meningeal dura, which sits on the actual arachnoid mater. So between those two, you have like a space. So that's where so between the arachnoid and the dura, you have a space, which is where the cerebral veins sit. So if you have a subdural hematoma, that's what people are talking about. And you have an extradural hematoma, that would be one that's outside of that's between the two dural layers, basically. So the next image I'll show you kind of makes that distinction a bit clearer. Then beneath that, you've got the arachnoid mater, and then after that, you've got the subarachnoid space. So the subarachnoid space is going to be where you have a lot of your bleeding if you have bleeding of one of the arteries around the circle of Willis. So then finally, you've got the pia mater, which is actually sits on the cerebral cortex and is kind of stuck to the actual brain itself. So this kind of makes what I was saying before clearer about which hematoma is where. This, I think, is also another quite a common MCQ question that comes up. So that's also something for you guys to have a look at as well. Yeah, so I will crack on. So now we're going to think about the actual brain itself. So when we think about the brain, it's useful to divide the brain into kind of three big parts. So there's going to be the forebrain, which can be divided into telencephalon and the diencephalon. We can talk about what all of these mean a bit later when we come to the neurodevelopment section. Then there's the midbrain, and then finally there's the hindbrain. So the other image that I have on the right shows the different parts of the different portions that comprise each of these different regions. I'm going to guess that all of you are quite familiar with these things. If not, like the images are there. So we're going to continue moving forwards. So I'm going to now just talk about the actual basic neuroanatomy and how the brain is sort of laid out. So apart from the forebrain, midbrain, hindbrain kind of division, we can also just think about the brain in terms of the cerebrum, the cerebellum, and the brainstem. So we're going to start off by thinking about the cerebrum. So when we think about the cerebrum, we want to remember that the gray matter, which is the neuronal bodies, are on the outside of the brain. Then the white matter, which is like the axons, are on the inside. So I think the easiest way to think about that is basically it's the opposite of what you the easiest way to think about that is so I personally remember that the corpus callosum is white matter and that sits on the inside. I don't know if that's helpful, but as that's just something that I'd heard of before we even came to medical school. So that's a way to remember it, I guess, if that helps you at all. So that's also quite another common thing that comes up in MCQs. So just for you guys to think about. So then in terms of the actual cerebrum itself, so the cerebrum is just another way of saying the cerebral cortex. So the cortex has lots of ridges called gyri and grooves called sulci. So the specific names for different sulci and gyri, we're not going to focus on those too much. But like I will talk about it on the next slide briefly, just so you're aware of some of the important ones. So when we think about the cerebrum, there are 5 key lobes that we can think about. So there's the frontal lobe, parietal lobe, temporal lobe, occipital lobe, and the insular cortex as well. So with the lobes of the brain, there's a little bit of like differences based on where you look. Some different things that I've seen suggest there's 6 lobes, considering the limbic lobe as another lobe, and some of them don't necessarily include the insula. For the purposes of this lecture, I've included the insula as well. So the insula is not visible on the outer surface. So if you kind of look beneath the parietal and the temporal lobe, you can see it here in this image where the green is. So moving forwards, this image kind of shows you the sort of key functions of these different lobes. I'm sure a lot of this is very familiar with all the essays and lectures and stuff you guys would have had. So a lot of the motor control and stuff is done in the frontal lobe. A lot of somatosensation is done in the parietal lobe. Occipital lobe is for vision. Temporal lobe is for auditory processing, as well as the site of the hippocampus. And then you've got the cerebellum for balance and coordination, and the brainstem for involuntary reflexes. I think just two key structures to remember is the precentral gyrus and the postcentral gyrus. So the precentral gyrus sits where you've got the central sulcus, which is just like here. So the one just before it is the precentral one. So that one is the motor cortex, or M1. And the postcentral gyrus, which sits immediately after it, is the somatosensory cortex, or S1. So those are just two very important gyri, which I think do get tested. A lot of the stuff otherwise is kind of more for interest, I think, in terms of like what exact gyri is what called what. But I think those two are important things that come up all the time. And they're kind of highlighted on the image to the right as well, if you look at it. So apart from that, when you think about the pre and post central gyri, we can think about this homunculus image. I'm sure all of you have seen it, but when I did second year, I didn't really understand it very well. But basically, it's an image showing which part of the brain is allowing motion, or which part of the brain is receiving the inputs from a particular area. So the size of the different parts of the body symbolize kind of the portion of the brain that's been dedicated to that. So if you look at this image, you can kind of see that the legs are relatively smaller in terms of how much area they're taking up compared to the face or the hand. So that's kind of in line with what we'd expect if you think about it, right? Because your hands and face are much more sensitive than your legs are, or your bum or something like that. And your tongue takes up a significant portion as well. So when you're thinking about like, this is also something that I think can come up in MCQs. It's definitely really important clinically, because if you think about having a stroke, for example, if someone has a stroke that's quite medial, so along the midline, their legs and things, their legs are going to be far more affected than their face or arms. And similarly, vice versa, if they have a stroke affecting the middle cerebral artery or something like that, that supplies the more lateral areas, then they're going to have those regions affected. So this is quite an important kind of image to have in your mind when you think about what is affected by a lesion somewhere in the brain. So then on the left as well, on the bottom left, we've got a picture of the hippocampus. So the hippocampus is involved with memory. The reason I've highlighted that is because of two reasons. So I think the hippocampus is one of the structures that I think they'd probably expect you to know exactly where it is. And also, I think you guys should recap like the limbic circuit and see how the hippocampus fits into that as well, because that's another common MCQ question. So another kind of thing for us to think about is the basal ganglia, but I'm not going to cover too much on that in this lecture, purely because I think there's a dedicated lecture to the basal ganglia that's coming up later. So just in terms of like revision and things, I'm not going to go through this image with you, but just remember that there's different angles of looking at the brain. So just have an awareness of what structures are where if you look at it from below or you take a sagittal section. I think just really important structures for you guys to be able to pick out would be the corpus callosum, which is the big white one in the middle. And I think you should also be able to pick out the thalamus, which is just like sort of beneath that, I think is the way I kind of remember that. So yeah, like the thalamus is there, and then you've also got the adhesion point of the thalamus as well. So if you just think about that, I think those are like some pretty important structures for you to be able to recognize on the sagittal section. Then in terms of the midbrain and the brainstem, so the midbrain, so the brainstem is composed of three different parts. There's the midbrain, the pons, and the medulla. So the midbrain allows motor coordination as well as visual auditory processing and arousal from sleep. So the anatomy of the midbrain is actually quite straightforward if you Google it specifically and try to look for it. So you've got the superior colliculus, which is involved with vision. You've got the inferior colliculus, which is involved with auditory processing. And then you've just got basically the bit above the pons. So the bit above the pons is kind of what does a lot of the arousal from sleep. So you've got lots of things that pass through the midbrain. So for example, the reticular activating system arises from around the midbrain area and allows you to be awake from sleep. So then you've got the pons, which is just beneath that. So the pons is really important for movement predominantly. So it also is what kind of connects the rest of your brain to the cerebellum. So there's different tracks within that go from the pons to the cerebellum that carry slightly different slightly different signals. So that'll be covered more in the cerebellum lecture. And then finally, beneath that, we've got the medulla oblongata. So the medulla oblongata is maybe the most important part of the brain for just keeping someone alive. It's what allows you to breathe, allows your heart to beat, and activates the GI system as well. So if anyone has a lesion of the actual medulla, they're more likely to die, and that will be a much more of a medical emergency than most other parts of the brain. However, it is important to remember that midbrain and pons are also really important to keep you alive as well. So the brainstem is kind of the bit of the brain that's important in just your basic functions that allow you to just kind of be alive. Whereas the cortex is kind of more complicated functions and more minutial things in a sense. So I think that's the kind of easy way of breaking it down and remembering what different parts of the brain do. So this is just another thing that I think comes up a lot in MCQs. I think it used to confuse me a lot when I was in second year, is what the different cross sections of the brainstem actually mean. So obviously the brainstem is a continuous structure. And even though like you can have differences between like the top of the midbrain to the bottom of the midbrain. However, I think these are like three different kind of like pretty standard images, I would say. And I think being able to know which one of those is which is really helpful. So the one on the left is the midbrain, the one in the middle is the pons, and then the one on the right is the medulla. So the way to remember the midbrain is the midbrain will contain the red nucleus. It will also have the colliculi. So if you look at the back and you see this like colliculi-like structure, that's a good way to remember that. And then you've also got the red nucleus as well. So that's probably, I think, the most straightforward way of knowing that you're in the midbrain. So then there is the pons. So the pons has this like thick layer of like fibers in the front. One of the people in the anatomy lab described it as looking a bit like a ball sack. So that might kind of also help you remember it as well. So I think with the pons, it is important to be aware though that there's quite a lot of variation from the top of the pons to the bottom. So not all of your images might look exactly like the one I have here. And then on the right, we have the medulla. So the medulla, the structure that I've always looked for with the medulla is the inferior olivary nucleus. You can also look for the pyramids as well. I think that's quite a good way of also being able to know which part of the brain you're in. So those are just like simple tips that I remember just as a way to like make sure that I know which bit of the brainstem I'm looking at. But by no means, like if you can pick out more structures, then obviously that's better and you won't go wrong. But I think, if you're struggling, I think if you remember like those three things, that will probably like cover you for 90% of the MCQ questions you're going to get about which part of the brainstem you're in. And then you can think about, what different structures mean as well. But yeah. So now we're going to briefly just talk about the ventricular system. So the ventricular system's basic structure was identified by Leonardo da Vinci in 1505. So he injected molten wax into the ventricles of ox brains and they formed casts. So based on those casts, they went on to kind of work out how the cerebrospinal fluid kind of flows around or flows in the actual brain. So the ventricular system contains the cerebrospinal fluid. So this is secreted by the ependymal cells in the choroid plexus. So these ependymal cells are one of the types of glia that kind of support the functions of the brain. So the CSF is really important because you've got your blood-brain barrier. That means that everything in the blood doesn't get to the brain. So this is because the brain is a really sensitive organ. And if you have different toxins and things going to the brain, that could potentially compromise its functions. So the CSF is providing nutrients, giving a stable environment, cushioning trauma, and also it allows you to remove metabolites quite quickly as well. So that's why we've got a separate designated kind of circulation to the brain that's not just regular blood like other organs. So that's the importance of actually having CSF. So in terms of how the CSF is flowing, you've got the lateral ventricles. From the lateral ventricles on the side, it goes into the third ventricle, into the fourth ventricle, and then finally you've got these granulations in the subarachnoid layer that allow the CSF to be drained away. So I'm just going to go back a slide, so like kind of be prepared for that in terms of your eyes. So if you go back to this one, so it's into the venous system in the subdural layer that the CSF is draining into through these arachnoid granulations. So I think someone said you can see my mouse, so that will be this layer here, I think. So this will be probably where you have some of your subarachnoid granulations, and then it'll diffuse into the sides. So hopefully that helps. Okay, so just in terms of some clinical significance for to be aware of, so hydrocephalus is basically where you have too much CSF in the brain. So that can be divided into two different types, communicating and non-communicating. So a communicating hydrocephalus is where you have excess CSF actually there. So you don't have an obstruction, you actually just have too much CSF either being produced or not enough of it is being absorbed. So you can have too much CSF being produced if you have like a tumor of these ependymal cells or something like that. However, that's incredibly uncommon. The most common reason of why you'll have a communicating hydrocephalus is that you're not actually absorbing the CSF normally. So you're quite likely to get that with a lot of conditions that affect the brain, really. Anything that can cause inflammation can potentially lead to a communicating hydrocephalus. So things like meningitis, things like inflammatory conditions like MS can also cause a communicating hydrocephalus. So then in terms of non-communicating hydrocephalus, those are essentially caused by obstruction. So you can have narrowing of different parts. So for example, a cerebral aqueduct constriction or like some kind of blockage from something else, a tumor potentially compressing the ventricular system so it doesn't drain properly. Once again, also inflammation can also cause compression from the outside. So all of those things, if you've got like a big bleed in the brain, so the blood can also cause a non-communicating hydrocephalus. You can also have lots of blood causing a communicating one if it actually bleeds into the ventricles itself. So if you kind of rip the thin layer that separates the ventricular system from the rest of the brain, then you can have bleeding into it, which causes a communicating hydrocephalus. So that's just clinical things for you to think about the ventricular system. So we're now on to kind of neurodevelopment. I know that we've covered like quite a lot of anatomy, and I know I didn't go into super detail. I just wanted to give you all like a basic principles and like think about different things rather than kind of get into all the minutial details. But if anyone has any specific questions, I have got the chat, so feel free to write it in the chat and I can try to answer it. But if not, we can just maybe move on. You're also very welcome to unmute and just ask any questions you have as well. Okay. So I'm guessing none of you have any questions about any of the neuroanatomy that we've talked about so far. If you have any more questions that come up, like you're very welcome to tell me as well. So now we're going to think about neurodevelopment. So the brain is a massively complicated structure. There's 100 billion neurons. We have about 20,000 genes to put that in perspective, you know, so there's just so many different neurons and there's so many different connections between them. And it's actually genuinely amazing that a structure like this can form so reliably across so many different humans. So when we think about neurodevelopment, like I said at the start of the lecture, I'm going to be focusing on kind of 4 key principles. But in terms of like the overall morphology, I think it's good to orientate ourselves. So on day 21 of embryological development, the basic structure of the brain has developed. We have a forebrain or a prosencephalon, we have a midbrain or mesencephalon, and a hindbrain or rhombencephalon. And then in week 5, the mesencephalon divides into the cranial telencephalon and the caudal diencephalon. I will tell you what those structures do in the next slide. And then the rhombencephalon or the hindbrain divides into the metencephalon and the myelencephalon. So once again, I'll tell you what those structures do. So the most important thing for you to remember is that the telencephalon is what goes on to become the cerebral cortex. So another like pretty, I would say, common kind of MCQ question is going to be like, they might give you something like, I don't know, the cerebellum, and they might say, which one of the brain vesicles has that developed from? You've got 5 options there, and you have to pick out the right one. I think I'm not going to spend too much time on this because I think this is just kind of like stuff that you can just learn. This is the diagram that I think kind of has all the answers on it. If you prefer tables, I've also got a table here that basically has exactly the same information. So from these basic brain vesicles that essentially are just swellings in the neural tube, you have these different derivatives, and I think this is a pretty like standard question. So once again, this is just something to flag for the MCQs. So now we're going to kind of think about it more in terms of the essays. So I've tried to incorporate quite a lot of experimental evidence. So you can put that into your essays to get the maximum marks. I also think breaking it down like this is actually quite a useful way of thinking about neural development rather than being like really focused on every little structure in the brain. So the first part that we're going to think about is early patterning in the brain and neurulation. So we're going to kind of skip over gastrulation. If you want to think about sort of the stuff that happens before neurulation, you can kind of look at what you did in first year, I think. So neurulation begins around week 3, 4, and it begins with the induction of a neural plate. So the neural plate is essentially it's composed of neuroepithelial cells rather than the less differentiated cells that you have in the initial bilaminar disc that you have at the end of gastrulation. So in order for neurulation to occur, you need to have an organizer, which is basically a particular part of the brain, or sorry, a particular part of the early embryological structure that allows you to form the actual neural plate and allow this change and differentiation in cells. So in humans, the organizer is the primitive node. If you're unsure about the primitive node, that's a structure that forms during early embryological development in gastrulation, I think. So you can maybe look over what that is if you're unsure. So the initial notion that you have an organizer was identified in the early 20th century by Spearman and Mangold. So they transplanted the organizer from one salamander embryo to another to show that it led to the formation of a full secondary axis, or the formation of basically a second nervous system. So this is a really important experiment, and it's kind of one of like the key experiments that's commonly cited when we think about neurulation. So then after you have the initial induction of the neural plate, you have selective proliferation of cells. So the cells begin to just divide, especially the cells on the neural fold, that causes the neural plate to bend and eventually fuse, allowing it to form into the neural tube. So this is kind of shown on the right in the images, where you have this convergence of the neural folds and they eventually fuse, forming your neural tube. So now we're going to think about the principles of patterning. So when we think about patterning, it's been studied really well in terms of dorsal ventral patterning rather than rostrocaudal. However, I'm sorry, I'm just moving certain things around. Okay. So in terms of the dorsal ventral patterning, there's two important signaling molecules. So you have on the ventral side, the notochord releasing sonic hedgehog, and on the dorsal side, you have the dorsal ectoderm releasing BMPs. So if you just skip over here, so you have basically BMP being released on one side, like at the top, just beneath the epidermis, and then on the other side, you have the notochord, which is releasing sonic hedgehog. So these different things have been proven by different experiments. So this is where you can bring lots of stuff in. So Holt-Fretter in 1933, I think, showed that when you transplant when you transplant a second notochord, you basically change the structures that form. So the sonic hedgehog is driving the formation of motor neurons in the spinal cord, and BMP is driving dorsal sensory neurons. I know we're not really talking about the spinal cord, but basically a similar kind of process is also happening in the level of the brain that allows the basic brain morphology and structures to be established. So on the other side, on the rostrocaudal side, so rostrocaudal is going to be basically this side is going to be rostral on one end, and the other side is going to be caudal. So on the rostrocaudal side, we have different signaling molecules. So we have retinoic acid caudally being released, and we have FGF being rostrally released. So these allow a kind of very similar process that essentially patterns the neural tube on the rostrocaudal axis, however. So I'm sure all of you have heard about Hox genes. I know that was something that was really emphasized in second year. So the retinoic acid is an important gradient that allows differential expression of Hox genes in order to allow you to have your different neuronal structures. So Coleman 1977 showed that if you change or disturb the retinoic acid signaling, then you have abnormalities or disruption of your normal Hox gene expression. So another really important principle when thinking about patterning, and especially early patterning, but also throughout neurological development, is cross-repressiveness within these morphogens. So when you have different combinations of different transcription factors, they basically specify a specific neuron to a position. So often these morphogens are repressing each other. So this is the example that's like quite commonly cited, I think. So you have FGF8, which promotes one particular gene called PAX6, and it represses another gene called EMX2. So these genes also mutually repress each other. So you have this like differential gradient with which I've tried to show with these like two triangles. So when you knock out one of them, what you get is you have basically expansion in some areas that are likely going to be induced by EMX2 and a reduction in other areas that are likely going to be induced by PAX6. So for example, when you knock out PAX6, you have expanded visual and auditory areas, but reduced motor and somatosensory areas. So this all kind of leads to this idea of a proto-map in the brain. So as I'm sure like all of you have maybe heard of at least, Brodmann basically divided the cerebral cortex into lots of little areas. Um, and basically the proto-map is this idea that early in development, the concentrations of these different morphogens interacting with each other, so at any given location, a specific combination of these morphogens is such that you have the development of different structures. So in line with that, there was an experiment by Rakic in 1988, which showed that you have different levels of transcription factors in different parts of the developing brain. So this diagram on the right is basically trying to symbolize that. So you have different levels of different genes being expressed in different parts of the brain that specify specific structures that go on to develop in those regions. Hopefully that's helpful. Okay. So then we can think about neuronal proliferation. So we've done our early patterning. Now we're on to proliferation. So the neuroepithelium is composed of these neuroepithelial cells that are pseudostratified in nature. So you have the cells that are closer to the inside, basically, where you're going to develop your CSF system. And then on the outside, you've got them that are the bits that are facing the meninges and the pia more broadly. So these pseudostratified neuroepithelial cells. So on the inner part is called the ventricular zone, and the outer part is the pial layer, basically. So the cells in the ventricular zone go on to proliferate rapidly to form neuronal stem cells as well as more differentiated neurons. So this next part kind of is definitely right side of the syllabus, thinking about things you can put in to get the most marks on the essays rather than kind of core stuff that's going to come up in your MCQs. So it's important to think about how these neurons are dividing. So the way to kind of think about it is whether they're dividing symmetrically or asymmetrically. So if they divide symmetrically, then you have the same levels of different proteins on both sides. And if they divide asymmetrically, then you can have an atypical distribution of the proteins, which is what allows you to define which neuron is which. So this mechanism of whether a division is symmetric to form more stem cells or asymmetric to form differentiated neurons is thought to be driven by the orientation of the cleavage plane. So the cleavage plane is basically the plane on which the cell is dividing. So if you have horizontal division, that's thought to be associated with an unequal protein distribution. So this was shown in this experiment that I've put here called by Chen et al. 1995. So basically what they showed is that this notch protein is located on one side of the cell. And as a result, if you divide horizontally, then more of the notch protein is going to stay on that side. And as a result, the cell that you've just divided is more differentiated than the one that had just previously been divided. Whereas a vertical division would mean that you have the same distribution of notch on both sides. And as a result, you'd have two cells that are identical and that remain stem cells. I think that can be slightly like a bit tricky to maybe understand. But if you Google some pictures, I think it really makes this point quite clearly. So then the mechanism of orientation, I think, remains unclear. So this is once again something for you guys to discuss in your essay. So there's this idea called interkinetic nuclear migration, which is basically where the nucleus moves on the central axis and kind of wiggles. And it's thought that maybe that's what's determining the plane of cleavage, essentially. And you can visualize this movement by staining the DNA. And there's a gene that's thought to be involved in this process. And if you impair that gene, which is what they did in the study, then you show that you've got a reduced number of neurons, and as a result, you've got microcephaly. So, this maybe supports the idea that interkinetic nuclear migration is what's driving whether you have symmetric or asymmetric divisions. So apart from the proliferation, it's also important that we have apoptosis in post-proliferation because you have lots of neurons being produced and only a few of them are actually needed in order to form your mature brain. So the importance of apoptosis was shown by this study by Kude et al. 2000, where they basically knocked out one of the caspases, which is what's driving apoptosis, to show the mice have enlarged brains. So then after that, we're going to talk about neuronal migration. So this is now we're on to our third key principle. So neurons migrate to establish the basic cytoarchitecture of the brain. So in the past, they used to think that the cells are kind of just dividing in the right position. But in reality, once you're past your very early stages of patterning, it seems that the neurons actually move to the outer layers and then subsequently get more differentiated. So this idea of migration was first shown in an experiment in 1961. So these two people injected radiographically labeled thymidine into cells. So this thymidine basically incorporated itself into the DNA of those cells. So then they would kill the animals and look at where the radioactivity was on different days. And they would show that the radioactivity, depending on when you injected it, would progressively get more superficial. And as a result, this suggested that the neurons were migrating. So the first type of migration that was identified was radial migration. So this was identified by Rakic in 1971. He showed that you have he basically showed that granule cells in the cerebellum migrate in order to on these Bergman fibers, which basically are some of the support structures in the cerebellum. In terms of the cortex, which is the main focus of this particular lecture, so more of the cerebellum will be covered on the cerebellum lecture. We know that the glutamatergic pyramidal neurons, so basically, the neurons that are associated with positive neuronal activity rather than the GABAergic ones, move on unbranched processes of radial glial cells. So once again, rather than the Bergman fibers, which are kind of like radial glial cells in the cerebellum, in the cortex, these are the radial glial cells, and they move from the inner ventricular zone out into the cortical plate and the cortical zone. So in terms of the mechanism of how they do this, this is a three-step process. Basically, the neuron wraps around the radial glia, then it transmits a force to move the centrosome forward, and then the centrosome pulls on the nucleus to create a saltatory movement, and this whole process of moving the nucleus is called nucleokinesis. So this is really reliant on myosin, but it's a different type of myosin to the one you have in muscle. So yeah, there's lots of different genes that are involved in this process. And if you complete the feedback at the end, a lot of the experimental evidence for these different genes and proteins are in the notes section of the actual PowerPoint. I'm not talking about it because a lot of it is kind of like they knocked out a particular gene and then that impaired the process in some to some capacity. So this is just a table that I made in second year with all of the different diseases that you can, or at least some of the different diseases you can have with impaired migration processes. So you've got the gene, what it actually does in terms of doing the movement, and the condition you can have if you have a mutation of this gene. It's just important to be aware, by the way, that all of these conditions are incredibly uncommon. So don't expect to kind of turn up to the hospital and see a lot of these conditions, but it's a good thing to understand these important physiological processes with though. So apart from radial migration, we also have another type of migration called tangential migration. So tangential migration occurs from three embryological structures that usually disappear after about 1 year after birth in humans. So these structures are called the medial, lateral, and caudal ganglionic eminences. So different neurons from these structures go on to become different interneurons. So interneurons is another way of saying GABA-releasing neurons in different parts of the brain. So those from the lateral ganglionic eminence go into the olfactory bulb, allowing us to smell. Those from the caudal ganglionic eminence go on to form cortical interneurons, and those from the medial ganglionic eminences go on to form Cajal-Retzia cells. So these cells are actually important in radial migration as well. And you've also got other interneuron populations in the basal ganglia. So it's just a destination of these different neurons, basically. So just as an idea of what these ganglionic eminences look like, we've got a picture as well showing how they sort of appear and where they're located roughly, and how the tangential movement works. So tangential migration is, as I said before, really important for the GABAergic neurons. So radial migration is for the glutamate releasing neurons in the cortex, and tangential migration is for the GABA neurons in the cortex. So if you knock out genes that are important in tangential migration, which is what they did in this Butt et al. 2008 paper, then you have a reduced number of GABAergic interneurons. So then in terms of the actual mechanism by which this happens, it's kind of still unclear, I think. But I think the basic idea is that because they're moving tangentially and not along another cell, it's thought that they are basically detecting some kind of chemoattractant, which is allowing them to grow in a particular direction. The actual movement itself is thought to be similar to radial migration driven by myosin. And if you block myosin using this particular drug, which is what they did in the study, this Bellion et al. 2005 study, then you can impair tangential migration. So those are the two types of migration that you need to be aware of. So the final principle that I talked about at the start of the lecture is this idea of integration of signals. So this is kind of a section that's maybe a little bit more wishy-washy in that it's got quite a lot of things for you to think about, but at the core of it, all of this allows you to integrate the different signals that you've established after creating different neurons located at different locations. So the first part is allowing specific growth of specific processes to make circuits. So this is thought to involve molecular matching, where basically you have a particular axon that's been made to grow towards a specific direction. So this idea of a chemo-specific hypothesis was tested by Sperry. This is one of the other really sort of classical experiments that often gets brought up in lectures. So I'm sure at least like some of you are quite familiar with this experiment. But basically, Sperry severed the optic nerve of a frog, and then he rotated the eye. So unlike humans, where our optic fibers don't regenerate like this, in frogs they do. However, what he noticed because he'd surgically rotated the eye was that there was an inversion of the behavioral reflexes. So it was just doing everything kind of backwards. And this kind of implied to him that the axons had connected to their original targets rather than where they needed to connect for the visual information to be correct. So based on this, he hypothesized that the axon was kind of growing towards a specific molecule that was being released. So a later experiment by a scientist called Bonhoeffer revealed that the specific molecule in this case is ephrin. However, that's more small print. It's more of the idea that you have a chemospecificity allowing axonal growth. So then in terms of other aspects for you to think about in terms of this integration of signals, we can think about transient circuits. So these transient circuits likely act as like a scaffold in the brain. There's lots of transient circuits that are intracortical as well, but the most important ones are, or at least the ones that have been studied a lot, are circuits in the subplate. So the subplate is one of the 6 layers that you have early in development. So just above the intermediate zone, you have the subplate if you look at this image here on the right. So these subplate circuits have been shown to be important in the formation of thalamocortical circuits. So what they've shown there's lots of different studies that support this, but basically in this Chen et al. 1988 study, for example, they showed that if you basically impair the normal development of these subplate circuits or you remove the subplate altogether, then the axons fail to grow normally to form your thalamocortical circuits. So maybe just in terms of your overall understanding as well. So the thalamus is like a relay center in the brain. So it's kind of what's connecting all of your sensory inputs to different parts of the cortex. So if you think about all of your special senses, whether it be like, whether it be sensation, vision, auditory, all of those things, you almost always pass through the thalamus before they reach the brain. So that's what I kind of mean when I've written thalamocortical circuits. So then finally, there's this idea of synaptic pruning. So this is also occurring throughout development. So I'm sure you've seen this like Hebbian theory, which is, those that fire together wire together, which is the circuits that get regularly used will kind of strengthen, and those connections are what become stronger. So the Hebbian theory is supported by like ideas of LTP, which is like long-term potentiation, and long-term repression, which is where you have where if you use a particular synaptic connection, then the connection actually grows stronger over time. So yeah. So then finally, just to end on, we're going to think about sort of when does neural development end? Because a lot of these processes are still happening early in development. So you have like a basic kind of timeline showing when these different processes are happening, allowing you to think about sort of what's going on when. So neurogenesis, however, is complete in adults, and that is important to remember. The only two parts of the brain where we still have new neurons being formed are the dentate gyrus of the hippocampus and the subventricular zone, which sends precursors to the olfactory bulb as well. Apart from that, we can still learn and change our neural circuits based on LTP and LDP, as I talked before. So those are concepts associated with learning that I think we have a separate lecture on memory and learning where they'll cover more of like the mechanisms of those two processes. But just for the purposes of this, I think just being aware that you can have changes in the strength of these connections is all that I think I really had to say on it. So that brings me to the end of the lecture. I'm just gonna, I know, well, I'll actually leave it up for a moment. So if I'm gonna stop recording.